flow cell that Scott and Jacob helped us to load and prepare. And then it sat there for 42 hours because I didn't come in to work the day after. I just worked from home on the Thursday, so I let it run. 42 hours. We got a lot of data out of this. This is not our flow cell, by the way. I didn't have the presence of mind to record it myself. My own nanoboard thing, but it basically just did that bit of flashing, a bit of popping on the screen for
More locked in than boomed on this screen.
42 hours. Okay.
You don't even have the slides on.
So what happened in that 42 hours? We had our DNA being unwound by the MOE protein and passed through the many thousands of pores on our flow cell. So we had about 1,500 pores that our DNA could pass through at any given time. As they pass through this membrane, they will trigger a change in the current, and each base gives this characteristic change. And once that has been recorded by the MinKnow software, which is the laptop had this software installed, it recorded the current. There's actually another pretty high performing software that needs to convert those signals into nucleotide bases and give us some quality information about them. So we took that data off the laptop, brought it upstairs to the fancy computer, which died. So we had to tweak that computer, turn it off and on again a few times, smack it on the side, and then it did what we wanted it to do eventually. So Perry Tobias worked really hard to get those bases called for us so we could have the data with us today. Okay, so we have the sequencing report that you can download here. So in the Canvas page, if you would like, or even if you've got these lecture notes open, that link should work. It should take you to the Google Drive folder that we had last week with our results, and you can download the sequencing report. It's an HTML file, which you can just you have to download it. You can't just click on it to view it because it will show up with some weird binary texty type stuff, but download the file and open it in your preferred Genome browser. So to download, usually for Google Drive, those three dots at the far right will be how you download, and then you can open that from the download folder and have a look. So you can see the general statistics. Has everyone managed to do the download and can open the file? We're going to be doing a lot of downloading from our Google Drive today, so don't close that folder. We'll be back there in a few minutes. So you should see something that looks a bit like this at the start. We have some general statistics, our data output, our base calling, our run duration, etc. And so this is what, this is just spinning out information. Our thing was run for 42 hours. We got over 12 gigabases of data and 18 million reads. Our N50 was 1.19. So I'll talk about that in a couple of slides, what N50 is. You probably already have heard that term before. We also have a lot of other charts. I'll just show you this one here. This is an example of how the pore health was looking over the time of the run. So as we started our run at the very first column, we can see that there are pretty close to 1,500 pores available. So when we did our pore check in the lab, we saw that we had 1,496 pores that were ready to go. So this matches that as well. Over time, those pores are getting depleted slowly as they do more sequencing. Some of them are just unavailable for a short while they're being, reloaded with a new fragment. And then there's some that just become inactive, and some that have zero activity. And this tends to increase over the duration of a run. So that's because the flow cell itself contains these proteins. They're actually proteins that are powered by normal cellular source of energy, ATP. And so the buffers that we put in our library have got all the solutions that are required, including ATP, and they get depleted over time. and the pores, the proteins themselves just get worn out and become less and less active. Okay? But we got a good amount of data in our 42 hours. Scott, would you reuse a cell like this with 400 active pores at the end? You've got something that after 42 hours we're sort of down here below 500. That's not sharing. Oh, I can see it on these screens. And your computer screen's not working over there?
I was looking at the wrong screen.
Oh, good. Yeah, would you use it all like that?
I would, depending on the experiment I had planned. So if you've got 400 pores left, you could just use it to like sequence a plasmid. If you want to, if your lab does that kind of work, you can definitely get some more juice out of something like that, or even for some metagenomics.
Cool. There you go. We're not going to reuse it. We just kind of put it in the pile for next year's class to do some sort of practice loading again. But yeah, you could potentially get some more data out of a flow cell like that. All right, so I wanted to talk about the read length chart that's present in that report. So if you have a look here, we have the length of the reads in kilobases along the bottom, and the number of bases that are represented by each read length. So when our report gave us an N50 value, and if you remember that from your sort of other genomics areas, we often use the N50 value to give us a guide for how complete a genome sequence is. So it is a measure of the contig length, and if you have an assembled genome sequence, the larger this N50 value is, gives you a strong indication that 50% of your genome sequence data is contained in longer contigs, which is what you want. And the ideal N50 value would be around the chromosome number, because you want your contigs, the size of your chromosomes, because you want your sequence data contigs to be assembled into as long as possible, and that is a chromosome. So for us, it's not actually that relevant because we've not done a genome sequence, but many people using Nanopore would be using this N50 value as a guide for how good their genome sequence is. Our guy from week 4 who did the ant genome would have taken an N50 value and said, yeah, I've got some really long contigs that I've assembled from my genome. For us, it doesn't really make that much sense because we did amplicon sequencing. So we had known fragment sizes already. All of our fragment sizes were about 1400 to 1600 base pairs in size. And this corresponds quite well to this nice peak we have here. So this is 1.2 to 1.65. That's about the size of where all our sequence amplicons should be fitting in. So our N50 value has said about 50% of the data occurs above this value here, 1.19. And that's where most of our amplicons are. What is this other stuff? It's probably some degraded samples, a bit of half reads that didn't get finished. Any other suggestions what the little short reads might be?
Yeah, potentially just things that have been dragged through the pore that are not what we're looking for, or that has broken throughout the process.
Yep.
For the longer stuff, you could get reads that have sort of attached to each other in some way, but they're going to get filtered out, or what you'll be doing shortly where you don't align to what you want, so we don't need to worry about those.
Yeah, okay, so thankfully we had a really nice peak where we thought we would. Yeah. Alright, so there's a few other reads that I wanted to, a few other charts that in that report. We're talking about data quality, so when you get some sequence data back, who's had sequence data back out of the demonstrators? Do you get your own sequence data, or do you use sort of published data sets mostly, or do you get your own sequence data back? Yeah. Yeah. So the first step I teach all my students is check your quality of your data, because any downstream analyses that you do, if you put in shit data, you'll get shit results. So you have to actually check the quality of your data, and you need to make sure that you only include data that's past a certain quality threshold. So we use this quality score called the Phred score, PHRED, H-R-E-D, not Fred, although couldn't help myself. I've got Fred here, Fred Flintstone. And the quality score is basically, if you have a score of 30, if your base score is 30, the chance that it is incorrect is 1 in 1,000. So, Fred is the original software that was used to determine the DNA sequences when the Human Genome Project began. So it's quite an old measure, but it's been maintained in the decades following. We often set a threshold to around 20. So that means you've got a 99% base call accuracy if your Fred score's around 20, but most often you're working with data that's 30 or above. However, if you have a quality that is below around 20, you might take some kind of software that trims off any of these poor reads. So your software will go, you set a threshold, trim anything that's less than 20, the Fred score is less than 20, because it's too high a chance and that base is just incorrect, it's bad quality data. We're not going to do any trimming today, just due to sheer weight of numbers. We have literally got tens of thousands of amplicon sequences, and so if we have a few reads that have got bad quality, they will be overwhelmed by the many reads that had perfectly accurate quality. Okay, so I want you to also have a look at two different quality comparisons of two types of data sets. And this is going back to what we discussed a few weeks back in week 4, where we were comparing long read sequencing with short read sequencing. I told you that often the quality of long reads is a lot lower than short reads, so that's one of the disadvantages of long read sequencing. And I want to show you this and how you would actually see that. So now go back to that results folder you opened before and download 2 more HTML files. These files were generated with a piece of software called FastQC. And they take FastQ files and they perform QC, and quality control. So whoever invented that name just ticks all my logic boxes. So have a look at each of these HTML files. I'll let you download those. I can see some downloading occurring. And just open those in your internet browser and have a look at what you see. You should see a couple of charts that pop up. I'm going to really focus on the first bar plot, so sort of box and whisker plot that you see of each. So you've got one for your nanopore barcode. So who did barcode 26? Do you remember? Whose was barcode 26? Must have been someone in the last bench, I think, because we were...
26.
Oh, last week when you were adding barcodes to your samples, yeah. Yeah, it's in the spreadsheet. We can look at that later. Anyway. Now this is not to name and shame, by the way. This is barcode 26, works super well. But we're looking now here at a box and whisker plot. So what this is showing us is this along the bottom is the position in the read, so the base pairs of the read. So the first 9 bases are labelled clearly, and then it kind of goes a bit logarithmic on us and jumps up to 1,500, 4,500, etc., increasing in size. So this is our nanopore sequence data. We've got reads that are up to 21,000 nucleotides, which is weird because we only put in, yeah, there should be there, but there's not many of those reads among them. Most of the data would be around this length here, so sort of maxed out at around 2 kb. So what we've got here is our mean quality in the blue, and it's sort of hovering around the 30 mark, which is pretty good, mean quality of 30. But we also see here, what a box and whisker plot shows you is the range. So you've got quite a large interquartile range here, where some of the reads positions have really high quality and some are pretty much dropping back down into the under 20 vicinity. So that's our nanopore data. Now what we have here is an Illumina sequence sample that I sent off for sequencing earlier this year. It's short read RNA sequencing data. Okay, so yeah, you know this sample, James? This could be one of the ones we worked on together. I want you to look at these two plots. If you haven't got HTML files loaded and working yet, don't worry so much. We're just going to be looking through these particular charts. What do you see that's different between the Nanopore one and the Illumina one? What are some obvious differences that have sort of popped up?
Low box and list of work.
Yeah. What's different about it?
So, with your range is maybe at 40 instead of 50 at the top, and mean low box and list is 30, and then mean is 40.
Awesome. So Will's just pointed out that our range is so much bigger in the Nanopore one. So it goes as high as 50 and like into the 10s. But the Illumina one is really very stable. It's right up there. That mean is at around just under 40 for the entire read. Speaking of read, what else is different? What else is different about the two sort of the base pairs of these two files? So we've got way different lengths. The lengths of our reads are completely different. If you look along the position in our Illumina data, it maxes out at 150, and that's because our data is 150 base pair reads. Our Nanopore is any length that it can happen to sequence on that given run. So that's our comparison. We get really variable Phred scores with Nanopore data, suggesting that the quality is highly variable across the reads. Our read length is huge compared to our Illumina data. Phred scores are consistently high, and our read lengths are all the same. Okay, so they're the two sort of things that you can gain from that plot. There's lots of other things we could work through in the FastQC file. Can't teach you all of them today. Probably something that you would get into if you were doing an honours project in next-gen sequencing, something that you would get to work through in your project. Have we had any questions that anyone's come up with so far? We're going to start our analysis shortly. Got a question? So this is the one thing that I don't like about FastQC is I don't give you a key for any of the plots. So we've got a really big, bright blue box here. So it's a quality one, and it's just, I think the fact that it's all blue is that the quality is all good. It's just quality. So sometimes you see sort of patchy white bits. Show me the Nanopore one.
Average quality.
Yeah. What is the
Where's the Nanopore one? Interesting.
How would I write a card?
Fantastic.
Why not?
Okay. So is this because our Illumina has some kind of different coding in the file that says it's got because of the tile? It's probably the way that the Illumina chip is sequenced. Yeah. Yep. So there are other different, nicely spotted. So our Illumina data is sequenced on a chip, a like a flow chip. They get attached to a chip, and that maybe that's triggers another quality measure that's called per tile quality. But the Nanopore does not have that measure.
Look this man in the face and tell him you don't want to do his Look his baby in the
Are there any other questions about quality? The take home, oh yeah, Jackie. Yeah.
It's kind of funny.
Well, yeah, presumably this is HTML.
It's possible that when we did the FastQC in that file, it was maxed out at 40, but it's also showing so that the Illumina one, it doesn't have any variation. So all the reads are just good. There's not like a range of reads that are a bit good and some that are better and some that are worse. They're all just really good. So it's also visible a range to the data.
What are we meant to be doing? I'm becoming a de facto group member.
Right.
What would you like me to do?
So, take home message. Phred scores. Basically, the words I have on this slide here is the Phred scores are much more variable and the read lengths are much longer in nanopore data. Whereas short read data, Phred scores consistently high, read length very consistent and the same. I'm glad to hear it. Yes. Okay, so let's get started on our own analysis. So what happened before you got here? As I said, we called the reads. They were demultiplexed into each of the individual barcode samples. So you all had your individual barcodes last week. We'll go through that spreadsheet again, so you can do a little bit of self-reflection on whether your samples worked or not. We had about, like I would say, over half of the samples that worked. And then there was quite a sort of some that kind of had a little bit of data, but not so much. And some that sort of had zero. So, well, I just thought we'd give you all a nice sweet treat when you arrive. That's your prize for just showing up, really. We don't have winners and losers in this class. We all get, everyone gets a prize.
Sometimes you catch fish and sometimes you don't.
Is that you? Oh, I don't want to. All right. So, Perry then went. So I don't know if you remember. Can I just have everyone's attention? Thank you. So Perry Tobias, who was in the start of the class last week, she took the data and she put it through that biosecurity pipeline. So this is a software package that they hand out to people working in governments who are trying to trace metal rust incursions, and they can say, just plug your data in here and it'll spit out a report for you. So you saw this file here that she gave us last week, and you would expect to get a green line if it matched the Australian strain and a red line if it didn't. So it's this automated analysis software that takes all of the 8 amplicons from your sample and performs a similarity analysis to the known strains. Now, what we discovered, because not every single sample worked, we were missing some of these amplicons. So there was not one sample that had the full set of amplicons that worked. There were some samples that had a high number of amplicons that worked. I think sample 2 and 3. But because some didn't work, we actually couldn't use this pipeline. It faltered. And so we learnt, and this is really, really helpful information for the biosecurity staff who are trying to perform this step, that you need a full data set for it to work. So what they're going to now go back and do is rewrite the pipeline to be a bit more robust, having some missing data. So Perry basically said, thank you guys, thank you for testing this, because it's really good to see what happens in the real world, because yes, thank you for screwing up some samples, because you've told us that we need our pipeline to be more robust to some missing data. And so they're going to go and fix that, and then they'll pass that on to people, end users. So awesome job, guys. Yes, absolutely. So why might some have not worked? Okay, there's a few reasons here. Obviously you had all these steps that we were doing. You could have failed at the A-tail step. This is where you're just taking an adenosine nucleotide, sticking it onto the end of the reads. If that doesn't work, then nothing else downstream works, because that really is the first step that you need to work. Now, I don't think anyone failed at this one here. I think all of our DNA concentrations were detectable. Every single person, every single sample had some DNA there. So you didn't lose your DNA. But if the DNA doesn't have the A-tail stuck on the end, it's not going to be barcoded properly. So there's a possibility that that A-tail step didn't work. Or there's a second possibility that the A-tail step worked and the barcodes didn't work. Maybe there wasn't enough ligase, maybe the conditions weren't optimal. That's sort of the two main areas where step 2 and step 5 would have possibly failed. So, based on that, instead of using that automated pipeline that Perry was going to show us, I've taken a different approach. I've selected a few successful samples that we're going to use for mapping and consensus sequence analysis. So I just wanted to escape from my slides for a moment and go to the spreadsheet. So here we have this, oh, I've got a few people already here. So I've highlighted the ones in yellow that I would say, for all intents and purposes, actually worked. So the ones in yellow worked. I've taken a sort of subset of these and kept some aside for analysis. So we could have done all of the ones, it's just that we have a lot of data and I just thought we'd get enough of an answer from the few samples that we have here.
Oh, let's go.
Okay?
No, you told them.
So we're now going to select a sample each, and you can pick the sample you set up yourself. Or you can choose any sample really. I just want there to be a range. I want people to have done each of the different ones. So if you set up, yeah, I mean like if you want to do your sample that you set up, fine, do that. Yeah, I want everyone to do their own sample if they've got it. If not, we'll randomly select. Okay, so as I mentioned, we wanted you to download Genius. Has everyone managed to go to the Genius website, www.genius.com, and download? So you will need to register that trial version with your email address. So if you've already used Genius before, you might need, as I said, to create a new email. So I'll give you 5 minutes to download Genius.
What does it say, Sophie? Sucked in?
And if you've already downloaded Genius, feel free to move on to step 2. Download a FastQ sample of your choice.
Sorry. I have the biggest ego in the world after you.
Wow, tell them to come over. I'm happy to dip whenever.
Able to reach the life of the
That's 5 words.
Oh, okay.
Well, my hot cutoff is 3 p.m. When is it? What time today? Oh, you've done it before.
Just going to hang out with each other. And everyone else. And everyone else.
Everyone else is like me.
The alcohol is not really good actually. We've got a few other facades.
You have become way more nice after that half a year. That's true.
I am not an alcoholic.
I can upgrade your trial, like.
Have you downloaded Genius?
Oh, yes. This.
Let's see if they want to.
No, I have a very solid password. I was considering voting for the ban Octoverse.
Just try to like console yourself there.
Oh, you have to download it.
That's very timetable.
I think it's, I got to that screen of well, but I already have it. Like, I've made an account, that's why I'm in here. Oh, but it is the free trial. No, but like, I just made my account. Activate my oh, it's really weird. I guess so. Schrodinger's. Thank you.
Oh yeah,
I love how at the top of Genius it says.
Can I please have a show of hands if you could download and install Genius?
Before you explain.
Has everyone managed to download it? Download. We're having issues getting the download installed. Good. Got it? Oh yeah, got a few more people waiting. Look, you can move ahead, but I would suggest just holding off until we're all caught up, just because I'm getting some questions which are coming on the next slide. So just pause. Okay, we can go to the next slide, but I want people to do step 2. So while you're downloading Genius, you can also start downloading your chosen FastQ file. So that could be the one that was yours in the spreadsheet. If yours didn't work, choose one maybe that was the same Applicon that you did, but a different sound.
No one can stop you from learning, guys.
Oh, Stan, what are you saying? What? Why is your first reaction to hearing someone got food poisoning to say made you feel like a troll?
What?
No, it is not a well-known thing. Yes, it is. Maybe in France land.
What have I missed on this chat? No, I knew that. I was like, what the fuck?
Wait, Tanya completely cooked you in a clapback. Oh my god, Stan.
After what my cycle push. Huh?
oh, so you've used this method?
Huh?
Yeah, I think that's
When do you want it?
well, you can download it.
I've given by 3 days. Well, 315
I read
315
shop
by the
it feel a bit better.
Stagger it. I'll wait outside.
Yeah, no, I'll wait outside.
Well, you want me to stay back? The 3 of you go? Yeah, and then I'll leave, and he will go. So then what do you call
Each one is a read. So each supplements and has your adapters. If this is your example.
Alright,
Yeah, it has
so
theirs, bucket,
Genius.
theirs, adapters. So
it is you all are. You are all geniuses.
I don't know about this.
It's a pretty good tool that I use every day if I'm having a good week. I'm in my happy place if I've opened Genius in any given day. It's a really intuitive and quite visual way to analyze sequencing data. You can perform many of the different steps that you guys have been already doing this semester via the command line, just in Genius itself. It has a lot of software preloaded into it. We're going to have to load one piece of software. So if you've gotten to that point, if you've skipped ahead, you might need to rein it back because I have one extra slide that I didn't include. So there is one caveat to Genius.
I got week 2 data.
It does not deal well with processing really large data sets. It's just because it's locally based. I think you can use a cloud version of Genius, but it doesn't deal very well with large data. So what you're often going to be doing is doing your command line processing that you've been doing in Linux and on a high performance server, like the virtual machine or something like that. And then you might get a BAM file output that you can then load and observe what's happening in that file in Genius. So it's kind of like really good for a small data set, but not for a really big one. So that's one thing to, so that's what I would normally do. I do my command line stuff, drag and drop the output into Genius to see what's happening. So what's the other thing I wanted to mention? All right, so if you have downloaded Genius, please choose your FastQ sample in the Nanopore data folder on your Google Drive.
What is time?
You can choose one. If you feel like going ahead and doing all of them, you are more than welcome to do multiple ones, but one that should suffice for today. Once you've downloaded your FastQ file, so this is the data, these are the sequences that came off the Nanopore. You need to also download a faster reference file that matches this amplicon that you've just downloaded. So say you've downloaded D3 barcode 15 FastQ, you'll need to go and find in this reference strain folder the faster file labeled amplicon D. So D3, that's amplicon D, that's your reference sequence that you want to map your reads to. Okay, so match your letters. If you've got A3 barcode blah, then download amplicon A, etc., etc. Okay. So who has done the downloading of the files, just so I know where we're at?
So we're just going to go through sample.
Okay. So people are ready to move on?
But there's only one.
Yep. So we want you to drag and drop these files that you just downloaded into your Genius window. Yep, fairly simple operation. Where did it download? Drag the file, drop it in.
Yeah, but the
Once you drag your FastQ file in, you're going to be prompted to tick some boxes. Now, we don't pair these files. If you're working with Illumina paired end sequence data, you would pair the files, but we don't, ours are not paired. I think. They're not, are they? No. Yeah. And then I also thought, why not, hey, we'll generate a QC report for each sequence. So click the box there and then click OK. That's the FastQC thing we mentioned earlier today. So you'll get to see the FastQC output of the file you've selected to analyze today. I should note here that these samples that I've given you from, I chose around 10, I think, of the successful ones. We had to make the file sizes a lot smaller. That's because we got a lot of sequence data. We got like thousands and thousands of reads. We've downsampled them. Some of them are about 5% of their original size. That's so we can handle the analysis in Genius. So once you've imported your FastQ, it will give you a few different tabs along the bottom. You can look at the sequence view, you can look at the length, text, etc. I want you to just have a quick scan of the QC report and you will be able to see the quality of your reads. Sorry, I saw some hands in the air. Are you?
Well, we're finished. It's a specific question.
Ah.
Download is each
Oh, that's the sequence report. So hang on, that's FastQ. They are. Oh, yeah. Go back to your downloads.
Okay, what's that? Got it?
I'll get back to you.
Oh yeah, so if your files aren't downloading from Drive, when was the last time you restarted your laptop? Because yeah, I find
What does that mean?
yeah.
Emily, if you ask me to restart my computer, it's like a 30-minute adventure.
Oh yeah, okay. Yeah.
I haven't.
That's fair enough. Okay, so it might be. You might just need a.
Oh, so you have to.
QR code. It's just I need to update my computer. Quick 15-minute. Well, it's kind of dependent. We'll see what happens. Sometimes it takes 5 minutes. Sometimes it takes an hour.
Alright, so you've got your sequences in. We probably should get moving so we don't take too long. So we need to install a little plugin for this mapping to work. So we want to map our reads that we've just imported, our FastQ file, and map it against the reference sequence. So you've done that before using BWA, I think a few workshops ago. You did some read mapping to a reference. Today we're going to use a different piece of software called Minimap2. You can actually use this via the command line. If we'd have installed it on the virtual machine, we would have done it that way, but we haven't. So we're now going to install it into Genius. So you have to go to the Tools toolbar, go to Plugins and select Minimap. Now, I didn't realize you had to do this until this morning when our lovely demos, Eloise, said, oh, Minimap's not in my thing. And I'm like, oh yeah, that's right. I installed that one for myself probably like a couple of years ago. So I've already installed it, but you just need to click on the install button and it shouldn't take too long to install. So that's the extra step that I have included.
It's interesting. I don't even know how it goes like this. I need to be connected to the internet to log into my computer.
Okay, so when that's installed, you can move on to the next step. So you want to select your FastQ file. So that's the one that has the barcode in its name. Tick that box next to that file. And then just above, there is a little button called Align Assemble. It gives you the option to do lots of different things. You could do a de novo assembly if you really wanted. We don't want to do that. We're just going to align it to a reference sequence. So we'll choose map to reference there. So when you map to the reference, you will need to choose your reference sequence. Now I'm not sure what will pop up for you. For me, I have to choose it because I have lots of different things that I've been doing in Genius. It might automatically come in for you. No, you've chosen your mapped reference. We then have a few other parameters. Has everyone managed to do that bit? Choose your reference sequence? Yeah, beautiful. So then you need to choose the mapper. So Minimap2 would be the one. Now, why do we use Minimap2 and not something else? Minimap2 is specifically designed for long reads. It's a new piece of analysis stuff that many people are using for nanopore sequencing. We use other short read mappers like BWA when the reads are short. So this has some sort of algorithm tweaks that mean it can handle the long reads mapping, probably some sort of different gap penalties and error things that change, so that the parameters are suiting the nanopore data set. I would suggest unticking including secondary alignments. So untick this box here. Don't trim. And I think everything else should be the same. You can save out an assembly report, and that report will just tell you how many reads in the amplicon actually mapped, and a few other things, and then save the contigs. So it's going to save the final output, and click OK. Are we all at the same stage? Oh, sorry. Need to restart your laptop.
Yes, there are.
Good work. Yeah, and it's just been like abandoned. You said it was the start on table of me. You did this in the fucking other lab as well. The point where Molly commented about it. What? Yeah, yesterday. Remember, like, first, second, I sat down first, and all of you went and sat down at the other bench. The other breakfast where I was set up as well. I'm not mad at it, but Molly came over to me and was like, oh, Will, you know you can, like, assist everybody else, right? See what
How's the mapping?
Yeah, I know.
Mapping away? Everything working? Alright. So, if your mapping has worked, you might see something that looks a bit like this on the screen. A large blue sort of bump and lots of black lines underneath it. So, I want you to see.
I don't know. I'm busy. I literally just did exactly the same thing. I think you decided to join us.
Right, so I can see some people have got their file. Still working on a few glitches, but we should have zoom into your lab reads. The next step is going to be generating your consensus sequence, right? So I think you've learned about this before. Have you learned about consensus sequences earlier in the semester? Yep. There are some yeses and nods. Because we have so many errors in our nanopore reads, we can't just take one read and go, this is my sequence, because there's a high chance there'll be an error along that read. So we need to use sheer weight of numbers to eliminate all these individual errors within each individual read and make a representative sequence of all the aligned reads. This will take into account the most common nucleotides at each site to give us our consensus. So you can do this in Genius pretty easily. You can select your assembled file. So you'll need to go to the one, sorry, my drop down bit has hidden it, but it's the one that has assembly in its title. Go up to the tools and all the way down to generate consensus sequence. For this, I'm just saying keep the defaults. I mean, I'm not sure what your defaults come up with. Is it similar to what I've got here, 60% highest quality? You can, yeah. The gap. I don't think it changes anything for the trimming, but we can try that. But yeah. The main thing is the threshold. We'll just keep it at the highest quality for now, but I will make a note here. There are so many ways to call consensus. James and I have probably gone through this round and round in circles on with our virus sequences. What's the best way to call a consensus? Is it about the quality of the reads? Is it about the just sheer number? Do we want to call heterozygotes? Because often when you're working with pathogen data especially, you're going to have a mixed pool. You're going to have lots of different genotypes altogether. So your consensus sequence reflects any inherent variation. You can actually get that variation captured by calling a heterozygote. I'm just going to say we'll leave it at our default setting with the caveat that in your future life as bioinformaticians or geneticists, you will have to choose the right parameters that suit your data. I don't want to say do it this way and do it this way every single time. It's totally dependent on what you're looking at. For our purposes today, it's fine for us to just use the default settings because we're just looking at amplicons, a PCR product. In your future life, you may change your parameters. So, we're going to choose our highest quality and generate the consensus sequence. It's going to write out a new file that will be the same name as this original assembled file and add consensus sequence to the end of it, just for your future reference. And so you've got your
pull up
the two files here. You've got your original sequence and you've got your barcode assembled. You want to align them to each other to see how they match. So we can use the muscle alignment. Muscle is just a different type of alignment software. I like Muscle, it's nice and fast. The Genius alignment I think is kind of is it slow? Yeah, Muscle is a bit faster. So we just use Muscle, it's perfectly fine. It creates pairwise alignment, it takes your reference sequence and your consensus sequence, matches them up, creates an alignment. Scroll along, have a look, zoom in and out. What does your alignment look like? You can see some stats on the left-hand side if you want to open up some of these. There's one that says percentages. It gives you the pairwise identity. You can see if there's any gaps, that sort of thing.
Boys are actually bad on touching the group shot. They're actually fucking bad.
What do you mean?
There is a missing picture. Someone tell me where it is.
Sophie, I deleted it just to piss you off.
It is until. Okay, you were right. It's encoded. It's not coding.
G support.
I still stand by what I said this morning. Yeah, because everyone hates this. I mean, the crocodile at the start. The crocodile at the start is no problem. Please.
Okay, so you've got your pairwise alignment. What this is actually showing you is how does our consensus sequence match with the Australian isolate. So AQ3 is our Australian reference sequence, and it matches pretty well. Now, my chats with Perry about data, I'm guessing you all have pretty good matches. Can you still want to yell out how well these are matching? So you can get a percentage match if you go along and look at your statistics. Got a 93 here. I had a 96. Lots of good matches. So if you just go to your right hand side of the alignment, there's some little tabs that you can look at, and there's a percentage.
Above 7.
Yep.
99.7.
99%, yeah. So we're really matching well to the Australian isolate. So, what is that actually? 90, yeah, 90. Yep. So that's telling us something. It's saying our sequence matches really well to the Australian isolate. So, this has kind of cracked the problem for our myrtle rust, because we're showing pretty convincingly that we have the strain that we thought we had. So we've not really had any new strains coming in. It's the pandemic biotype that we thought we would have, and that is pretty good news. We don't need to send any reports to Plant Health Australia to tell them we've got a really weird new exotic strain coming in. But to be completely sure, I think rather than just looking at the similarity, you really need to look at more reference sequences from around the world. And so we have some additional reference strains that we'd like you to download now. More downloading from Google Drive. So if you've been working with a particular amplicon, stick with the same one here. It's just more sequences of the same thing. Lots of different references, maybe 6 or more in one FASTA file. So download this one. Now, when you import it, we want want you to create a list because it's keeping, you're right behind me, it's great. You want to keep everything in a list so you don't end up with 10 different sequences on separate lines. Okay, so drag and drop your new FASTA file into Genius. And we're going to do the same thing now, but I want you to select your new list of sequences, your amplicon list, and your consensus sequence. And when you go to align or assemble them now, it doesn't ask for a pairwise alignment, it says multiple align because you have multiple sequences to align. So you just do the same thing, click on muscle. No, so the original AU one is actually in that multi-ref list. Yeah, it's going to be there anyway. And we get a multiple sequence alignment. So you should have maybe 6 or more different strains in there. Yeah, so some of those similarities will reduce now because you've got more sequences in the data. You've got some from Brazil. So we've got our AU strains. And I think the MF1 strain is a Brazilian isolate. I'm not sure, I think the others are from South America too, which is the host local, where the metal rust originated from. And then you can see the alignment, there's some similarities and differences across that amplicon. But a really simple way to determine how similar they are against each other is to generate a quick tree. Now, I realize this is not the right way to teach you phylogenetics because this is a really limited data set and there are a lot of different steps involved in making a good phylogenetic tree. But I often use a tree to help summarize data in a quick and easy way. So when you're looking at an alignment of 1,000 bases and you're trying to say which one is more like the other, it's really hard to tell that by just looking at the nucleotides. So you can get a tree building software to just basically tell you the similarities for you. So it's a visual way of computing how many similarities and differences there are between each of the multiple sequences in your alignment. So you can take your nucleotide alignment, this one containing all the different references, and then just choose tree, and we can just use a default Genius tree builder. It's a quick way to just make trees, basically. If you want to do some more hardcore phylogenetics, you probably should do the evolutionary biology unit in first semester. You've probably been shown how to make a real tree in that unit, I'm guessing. Yeah. But for today, that's all we need. We just need a quick tree. And then you can look and see what your consensus sequence matches to the most. My suspicion is that everyone in this room should have a high match to the Australian isolates, so things that have AU3 next to it. I did have a quick look at some of these amplicons, and all the ones I checked were matching to Australia. So, and I don't know what the asterisk is.
Alright, I'll report some notes for it.
Alright, so if you feel like doing more, you can go through and do another amplicon. But because the class has pretty much done them all, we're pretty happy there. I just want to ask a favour and give a thanks. What do we call it?
What?
So, I want to thank you for your involvement in testing out this protocol.
Okay.
We've shown that it works.
Yay.
We've shown that we can detect new isolates, though we have learned that it's best if all the amplicons get sequenced, because then the automated pipeline will be more forthcoming. But we can still analyse the data in the way we have today. And in fact, I think this is a bit more of a hands-on approach. You're actually getting to see the sequences, you're performing the alignments, you're looking at where, what they're matching to. So I wanted to thank you all for getting involved and helping us out with that. I have a favour now, after I've thanked you and applied you with chocolate. I would very much appreciate your feedback on the last 2 weeks, because this is the first time we've run the lab. And if, it's been great, but it's a lot of work and actually rather expensive. So if we want to keep running it, it has to be something that we know is valuable to you and has helped you to learn. So I've put together this very short survey. It would not be a class with me if I didn't have a Menti link, really, would it? So if you could give me some feedback on this class. I really am keen to see if it was valuable, if you learned something, if you enjoyed it, and whether we should do it again. Demos, you can fill out the survey too. Because all of this is really good feedback. I think, some things I have learned about what we could do differently. I think we had a bit of downtime in the lab. I'm not sure that was the most productive way to spend your time. So we could have had sort of a mini lecture while you were waiting for the library in the lab. We could have got you to come back an hour later, that sort of thing. I liked that it was an authentic question on real samples that had been collected weeks earlier. I don't know, I'm just wanting to hear your feedback and your opinions on what you liked and didn't like about it. And we'll work with that. But I hope it was something you valued and enjoyed, and I was very happy to do that with you. We're going to stick around until you need us if you have questions. I did want to remind everyone about the Honours Information session at 4 o'clock today over in Edward 23. So everyone's going to the Honours Info session. Yeah. Good. So, if you want to stick around in here, feel free. Any questions? How's our chance? Yeah. I'm going to the Honours, so...
Hi.
Where's the other guys here too?
What?
Where's the other guys here too?
I don't know. They just went out one by one. So.
One by one.
uh,
I joined the group too.
Group 2. Okay, wait.
I think we can just wait to see if there's anyone who's in the discussion.
Yeah. Let me join in.